We can continue. Which of the following macronutrients require the most energy for assimilation? Uh, because proteins are more the thermogenic property is more, so most of the energy for it requires more energy for assimilation. So among the macronutrients, what are the macronutrients? Carbohydrate, protein, fat, right? So among these, proteins require most energy for assimilation. Which of the following methods cannot be used to precipitate protein? Alcohol, acetone, heavy metals can be used. Trichloroacetic acid can be used. But this isoelectric pH is not a method for precipitation of protein. What is the shared precursor amino acid for urea, creatinine, nitric oxide? Arginine. From arginine, nitric acid is being formed. Creatinine is formed from arginine, methionine, glycine, arginine, methionine. Then urea is formed from arginine. Okay. Then what are the following is which of the following is responsible for a negative charge of fibrinopeptide A? Negative charge of fibrinopeptide A is by aspartate and glutamate. Fibrinopeptide A contains aspartate and glutamate, which provide the negative charge for fibrinopeptide A. What is the reason behind the stability of adenosine receptors, which is attributed to the numerous disulfide? Disulfide அப்படின்னாவே sulfur containing amino acid தான் நீங்க select பண்ணும். But cysteine is there, methionine is there. But adenosine receptors வந்து cysteine தான் attribute பண்ணுது. The stability of the adenosine receptor is mainly by cysteine. Then biochemical form of amyloid and hemodialysis associated amyloidosis is beta 2 microglobulin. Beta 2 microglobulin. I think you have read in your pathology and medicine. Bodybuilder starts eating raw eggs for protein. He starts to develop fatigue on moderate exercise. The doctor prescribes a vitamin. Which vitamin is deficient in him? Pyruvate carboxylase. Pyruvate carboxylase. Thiamine. Okay. Thiamine is responsible for carboxylation reaction. Sorry, biotin. Sorry. Biotin for carboxylation reaction. Okay. This egg contains biotin. Okay. So this patients will be deficient, starts prescribing a vitamin known as biotin. Okay, because pyruvate carboxylase is deficient in him. Biotin always causes carboxylation, mediates a carboxylation reaction. Okay, when which of the following is not requirement for activity of DNA polymerase during replication? During replication, what's not required for this one? It needs primer. DNA replication DNA replication, DNA polymerase needs primer. It needs DNA template from 3 prime to 5 prime end, because synthesis always from 5 prime to 3 prime direction. So it needs 3 prime to 5 prime DNA template, and the primer should be needed for the DNA polymerase, and magnesium will be responsible for the polymerase reaction. But it is not required, but it needs the nucleotide triphosphate. DNA polymerase will act only with the presence of these 5 prime to 3 prime to 5 prime template, primer, then magnesium. Then only it activates. Then what is the wavelength at which nucleic acid show maximum absorbance of UV light? 260 nanometer. 260 nanometer is the maximum absorbance of UV wavelength, UV light for nucleic acid. What sequence is repeated in human telomere? 5 prime to 3 prime, TTA, GGG. 5 prime to 3 prime, TTA, GGG. This is the sequence repeated in human telomere. Which of the following is not caused by defective nucleotide excision repair mechanism? Burner syndrome is not is caused by this. These three will be caused by defective nucleotide excision repair, like xeroderma pigmentosum, Cockayne syndrome, trichothiodystrophy. What is the role of UVR ABC exonuclease in nucleotide excision repair? It recognizes and excises the damaged DNA segments. Exonuclease means it excises and remove the damaged DNA nuclease. Mismatch repair defects are mostly associated with hereditary non-polyposis colon cancer syndrome. Mismatch repair defects, hereditary non-polyposis colon cancer syndrome. If you read DNA repair mechanism and read this MCQs, you will remember. Why are methylases important in mismatched repair mechanism in prokaryotes like E. coli? Because they methylate the parental DNA strand, allowing mud proteins to distinguish it from daughter strand. ஸோ மெத்திலேசஸ் வந்து நான் என்ன பண்ணோம் அப்படின்னா பேரண்ட் ஸ்டாண்டில் இருக்கிற டிஎன்ஏ ஸ்டாண்டை மெத்திலேட் பண்ணிட்டு அதை வந்து டாட்டர் ஸ்டாண்ட்லேருந்து டிஃப்ரென்ஷியேட் பண்ணுறதுக்காக தான் இந்த மெத்திலேஷன் வில் ஹேப்பன். விச் ஆஃப் த என்சைம் ஹெல்ப் ரிமூவ் த டிஃபெக்டிவ் பேஸ் டியூரிங் எக்ஸிஷன் பேஸ் பேர் எக்ஸிஷன் டிஎன்ஏ கிளைகோசிலேஸ். டிஎன்ஏ கிளைகோசிலேஸ் என்ன பண்ணும்? இட் ரிமூவ் த டிஃபெக்டிவ் பேஸ் டியூரிங் பேஸ் எக்ஸிஷன் ரிப்பேர். ஓகேவா? அடுத்தது 6-year-old child presents with recurrent respiratory infections and poor growth despite normal diet. Genetic testing reveals cystic fibrosis. What type of mutation causes cystic fibrosis? Allelic heterogeneity. Allelic heterogeneity. Then, which of the following statement accurately describes the role of DNA polymerase in eukaryotic cell? DNA polymerase are of eukaryotes are of 1, 2, 3, 4 and 5. Okay. So the three mainly responsible for the polymerization reaction in eukaryotic reaction. It requires primer and it provides proofreading during leading and lagging strand. Okay. For synthesis of DNA. Replication. In replication, what will happen? Template is there. 3' to 5' template is there. It synthesizes 5' to 3' direction daughter strand. It requires RNA primer. RNA primer is required, and also it will do proofreading mechanism like during lagging strand. Leading strand also. Leading strand will be continuous. Lagging strand will be semi-discontinuous. These are all Okazaki fragments. Okay. Which of the following statement about telomere and telomerase is correct? Telomeres are composed of repetitive sequence that help prevent chromosome fusion. There will be repetitive sequences there. So there will be prevention of chromosome fusion. Okay. Then which of the following statement about DNA polymerase in E. coli is true? DNA polymerase 3 has the highest rate of chain elongation and is involved in leading and lagging. I told you, no, type 3 is responsible in eukaryotes. Prokaryotes la vandu alpha, beta, gamma, epsilon. So gamma will be alpha, beta, gamma. Third part. Third is the responsible for polymerization of the DNA. So DNA polymerase 3 has the highest rate of chain elongation and leading and both leading and lagging strand. Which of the following is not required for DNA replication in eukaryotes? Eukaryotes la vandu RNA polymerase is not needed. Only DNA polymerase is needed. What are the other requirements? Origin of replication, DNA helicase to unwind the double strand. RNA primer is responsible for the action of the DNA polymerase. Which of the following disorders are not caused due to defect in non-homologous end joining? Not caused. So what are the things which are caused? Ataxia telangiectasia, Bloom syndrome, severe combined immunodeficiency disorder. So non-homologous end joining defect is defect நால வர diseases என்னன்னா ABC, that is ABC, ataxia telangiectasia, Bloom syndrome, severe combined immunodeficiency disorder. 45-year-old male women present with visual disturbance, vision loss, difficult seeing in low light, along with muscle weakness and fatigue, early onset cardiac conduction defect, worsening symptoms over the years. Her family history shows the similar symptoms in her mother. Genetic condition best explains her symptoms are Leber's hereditary optic neuropathy. So visual disturbances is there, 45-year-old male, and run in families. So it is hereditary optic neuropathy, mitochondrial disorder. Okay. So the following is characteristic of mitochondrial DNA. It has very few non-coding region, and higher mutation rate compared to nuclear DNA. It has non-coding region is very few, mutation rate is very high compared to nuclear DNA. What is the role of inosine monophosphate in cellular metabolism? It acts as a precursor for adenosine monophosphate and guanosine monophosphate. Inosine monophosphate From inosine monophosphate only, guanosine monophosphate and adenosine monophosphate will be formed. What type of bond forms between nitrogenous base and pentose sugar in nucleoside? Beta N glycosidic bond. N glycosidic bond is correct, but compared to alpha and beta, beta N glycosidic bond is the bond which connects the nitrogenous base and pentose sugar in nucleoside. Which of the following is not caused by double-stranded DNA repair? Coccini syndrome. So what are they? Fanconi's anemia. I told you, no, this non-homologous end joining disorders, ataxia, Bloom's, severe combined immunodeficiency, same thing. That is DNA repair defect is due to Fanconi's, severe combined and Bloom syndrome. Then what are the following statements about homologous DNA repair is correct? Homologous repair uses homologous DNA template for repair, making it highly accurate. It will not use heterologous or non-homologous DNA template. It uses only homologous DNA template for repair, making it highly accurate. Okay, what is this? Origin the DNA, this is this, mutated is this. Here it is UAA. What is UAA? Stop codon. Stop codon is UAA. You will be asked, UAA is stop codon, start codon, non-codon, exons, introns, likewise they might ask. So, stop codon. So, in mutated sequence, there will be stop codon. There will be premature stop codon. Premature stop codon, which is known as nonsense mutation. Okay. அதே mutation இருக்கு ஆனா final stop codon is there. So, it results in nonsense mutation. Okay. Identify this type. Identify the type of mutation illustrated in the following DNA sequence. Starting sequence, mutated sequence. See here, this will be deleted. This is deleted. So, deletion mutation occurs if it is deleted. This function of the gene will be altered. There will be mutated gene. Okay. Then, which technique is commonly used for DNA sequencing? To identify mutation by determining the precise order of nucleotide in DNA sample. Fluorescence in situ hybridization. That is FISH. Fluorescence in situ hybridization. So, DNA sequencing வந்து யூஸ் பண்ணி மியூட்டேஷனை ஐடென்டிஃபை பண்ணுற மெத்தட் என்ன அப்படின்னா fluorescence in situ hybridization. To identify the mutation, DNA sequencing method என்ன மெத்தட்னா fluorescence in situ hybridization. Sickle cell anemia. Sickle cell anemia என்ன ஆகும்? Sixth position of the beta globin of the hemoglobin வந்து glutamic acid is replaced by valine. GV பிரகாஷன் ஞாபகம் வச்சுக்கோங்க. Sixth positionல GV பிரகாஷ். Glutamic acid is replaced by valine. Okay va? So this is missense mutation. So because of this, the normal hemoglobin will be lost. So sickle cell anemia will manifest. It is mediated by missense mutation. Okay va? அடுத்தது, which of the following best describes a mechanism involved in epigenetic changes that lead to heterochromatin formation? What is heterochromatin? What is euchromatin? Heterochromatin means loosely arranged cells, so genes will be expressed. Euchromatin means tightly arranged cells, so genes cannot be expressed. Okay, so transformation of euchromatin to heterochromatin is known as chromatin remodeling. So this heterochromatin formation needs histone deacetylation and methylation in CG islands. Deacetylation, acetylation கிடையாது. Heterochromatinனா லூசா இருந்தா தான் genes will be expressed. Chromatinனா protein which surrounds the either, so only DNA will occupy the cell. So DNA deacetylation and methylation. In CRISPR, what is CRISPR? Clustered regularly interspaced short palindromic repeats. That is CRISPR. Cas9 experiment with mammalian cell line. The research team observed repair of DNA double helix pattern. Which repair are most likely observing? இந்த CRISPRல வந்து எது வந்து most likely observing? NHEJ, that is non-homologous end joining and homologous end joining தான் வந்து repair mechanism நடந்து அதை வந்து correct பண்றது. That is clustered regularly interspaced with short palindromic repeats. இதை வந்து in vitroவா பண்ணும் போது இந்த ரெண்டு மெத்தட்ல வந்து பண்ணாங்க. ஓகேவா? These two mechanisms are most likely observing non-homologous and end joining and homologous end joining. which of the following is a feature of M phase? M phase na enna? Mitotic phase. Okay, இந்த mitotic phase ல வந்து PTAM. What is P? Prophase, PMAT, sorry, prophase, metaphase, anaphase, telophase. So இந்த metaphase, prophase, metaphase, anaphase, telophase ல வந்து இதுல ஒரு phase ல வந்து chromosomes are highly condensed to facilitate segregation. ஓகேவா? S phase ல தான் வந்து DNA replication வரும். synthesis phaseம் வந்து S phase ல தான் வரும். ஓகேவா? ரெப்ளிகேஷன் ரிலேட்டடாக எல்லாமே எஸ் ஃபேஸில் தான் வரும். ஸோ இந்த மைட்டோட்டிக் ஃபேஸில் தான் வந்து ப்ரோஃபேஸ், மெட்டாஃபேஸ், அனாஃபேஸ், டெலோஃபேஸில் தான் குரோமோசோம்ஸ் ஆர் ஹைலி கண்டென்ஸ் டு ஃபெசிலிட்டேட் செக்ரிகேஷன். 32-year-old patient present with family history of progressive muscle weakness, decline, involuntary, microsatellite instability, CAG repeats. ஓகே, எது வரும்? Duchenne muscular dystrophy. Okay, CAG repeats will be there. Okay, CAG repeats will be there. This is Duchenne muscular dystrophy. This is I will clarify and tell. Okay, 5-year-old boy, finally I will tell. Okay, this for this question. 5-year-old boy presents with frequent falls. This only Duchenne muscular dystrophy. Frequent falls. 5-year-old boy. Difficulty in climbing. Unusual way of getting up from the floor. Genetic testing வந்து muscle disorder இருக்கு. இந்த involved gene வந்து dystrophin gene. So this Duchenne muscular dystrophy ல தான் வந்து இந்த presentation இருக்கும். இது வந்து it is what is which is true in this largest in the human genome. Dystrophin gene is largest in the human genome. ஓகேவா? Satellite DNA has various applications due to its unique repetitive sequence. Which of the following is not application of satellite DNA? இந்த satellite DNA அந்த repetitive sequence வச்சு இந்த DNA வந்து செக் பண்றதுக்கு வந்து அதோட அதோட வாட் ஆர் த யூசஸ்னா கிரிமினல் இன்வெஸ்டிகேஷன், ஃபாரன்சிக் ஐடென்டிஃபிகேஷன், பெட்டர்னல் டிஸ்பியூட், ஜெனட்டிக் டைவர்சிட்டி இதுக்கெல்லாம் யூஸ் ஆகும். பட் நாட் கார்டியோவாஸ்குலர் சசப்டபிலிட்டி இருக்கா, டிசீஸ் சசப்டபிலிட்டி இருக்கான்றதை வந்து கண்டுபிடிக்க முடியாது. The following statement is not accurate regarding post-transcriptional modification of eukaryotic RNA. Eukaryotic RNA ல என்ன ஆகும்? Addition of poly A tail at 3' end, methylation at the 5' end, அப்புறம் removal of the introns and the exons will be added. இதுல வந்து ஸ்ப்ளைஸ்டுன்னு போட்டிருக்காங்க. introns removed, exons are spliced கிடையாது. introns are removed and exons others will get joined. ஓகேவா? இட்ஸ் நாட் அக்யூரேட் இது இந்த ஆன்சர். மற்றதெல்லாம் ஓகே. மெத்திலேஷன் அட் 5' end, capping at addition poly A tail at 3' end, 5' capping at 5' end. ஓகேவா? அடுத்தது researcher aims to clone a large eukaryotic gene fragment for use in functional genomic studies. Which of the following cloning vector is the most appropriate for this purpose? large eukaryotic gene fragment வந்து clone பண்றதுக்கு வந்து yeast artificial chromosome தான் தேவை. 600 kb அப்படின்னா yeast artificial chromosome தேவை. In human somatic cell, which of the following statement accurately describes the chromatin and its characteristics? Facultative heterochromatin includes bar body. இதுதான் வந்து chromatin describe பண்ணும் somatic cell. Bar body, heterochromatin. In laboratory setting, E. coli bacteria are cultured in medium where glucose and lactose is available. To increase the expression of the lac operon, which level of lactose and cyclic AMP would you expect to present? லாக்டோஸ் ஹையாக இருக்கணும், ஹை சைக்ளிக் ஏஎம்பி இருந்தால் தான் எக்ஸ்பிரஷன் வந்து அதிகமாக இருக்கும். ஓகேவா? ஃபார் இன் ஜீன் எக்ஸ்பிரஷன் இன் ப்ரோகேரியட்ஸில் ஹை லாக்டோஸ் அண்ட் ஹை சைக்ளிக் ஏஎம்பி. Which of the following technique is specifically designed to introduce genetic modifications such as gene knock-ins and gene knockouts into genome of organism? வந்து CRISPR-Cas. அந்த இன்விட்ரோ சொன்னோம் இல்லையா? இன்விட்ரோ சொன்னல இதுக்கு முன்னாடி CRISPR டெக்னிக்ல in homologous end joining and non-homologous end joining. அதே தான் இதுவும். Okay. What is this? This is microarray technology. What is the role? It is used to study multiple gene expressions simultaneously. Multiple gene expressions simultaneously. Microchip இருக்கும் நிறையா. Simultaneous we can read many multiple gene expressions. So the following vectors is most suitable for planning large 300 kb na bacterial yeast chromosome, bacterial artificial chromosome. 600 kb na yeast artificial chromosome. Okay, yeast na 600 kb, bacterial na 300. 300 வந்து prokaryotes, அதனால 300 kb. Yeast வந்து fungus 600 kb. Okay. Which type of blotting technique is used to specific RNA? Northern blotting. Northern R இருக்கு இல்லையா, அதனால RNA. Southern blottingனா DNA. Western blotனா protein. அடுத்தது, which type of restriction endonuclease creates staggered cuts in DNA producing sticky ends? Staggered cuts and sticky ends na type 2 restriction enzymes. Type 2 restriction enzymes. Child present with micronathia and low-set-ears, these features are commonly associated with which type of genetic abnormality? வந்து Cry-du-Chat syndrome. இது நான் உங்களுக்கு சொல்றதுக்காக எழுதியிருக்கேன். மைக்ரோனெத்தியா லோசெட்டியர்ஸ்லாம் இருக்கு அப்படின்னா Cry-du-Chat syndrome, deletion of 5p, short arm of p na puny, short arm of p chromosome p. What is true about Hayflick limit? Hayflick limit na வந்து human cells வந்து can divide a finite number of times only. அதாவது 40 to 60 times only they will divide. So only aging process will come. இப்போ நீ வந்து 20 இயர்ஸ்ல பார்க்கறதுக்கும் 60 இயர்ஸ் 70 இயர்ஸ்ல பார்க்கறதுக்கும் there will be finite number of division will be only finite times, 50 times a division, 60 times a division. That's all. Up to that times only this cells will divide. More than that they will not, because aging will aging process வந்து இது வந்து depict பண்ணுது Hayflick limit. ஓகேவா? Which of the following is not associated with post transcriptional modification? I told you methylation at 5 prime end, capping at 5 prime end, endonuclease activity உள்ள வந்து intronsலாம் கட் பண்ணி எக்ஸான்ஸை வந்து கொட்ட வச்சு glycosylation மட்டும் நடக்காதாங்க. Glycosylation is not at all linked with this post-transcriptional modification. Which type of mutation results in reversal to wild type of phenotype when the mutant gene is expressed? Mutation in tRNA. Mutation in tRNA ல தான் வந்து wild type phenotype க்கு வந்து reverse ஆகும். Fish technique help to identify all except. Fish technique மூலமா translocation, deletion, gene amplification எல்லாம் பண்ணலாம். ஆனால் point mutation identify பண்ண முடியாது. அடுத்தது, which of the following uses CRISPR-Cas9 technology, DNA editing by homologous end joining and non-homologous end joining? Okay. RNA editing எதுக்கு சொன்ன? அப்போ B8, B50, 48, B100ல இருந்து B48 intestineல நடக்கும் by means of RNA editing in intestine. ஓகேவா? DNA editing வந்து this uses CRISPR-Cas9 technology. Which of the following post-transcriptional modification is common to collagen, hemoglobin, immunoglobulin? Subunit aggregation, that is alpha, beta, to alpha, to beta. So subunit segregation, post-transcription modification. இந்த இதுல வந்து என்ன type of post-transcription modification இருக்குன்னா subunit aggregation. 7-year-old child of short stature presented to outpatient department with skin patches and learning difficulties. Diagnostic workup, they show defective DNA repair due to double stranded breaks. What is the Bloom syndrome? ஓகேவா? ஸ்கின் பேச்சஸ் இருந்தா ப்ளூம் சிண்ட்ரோம், லேர்னிங் டிஃபிகல்ட்டியோட. இப்போ 13-year-old with flaccid paralysis, RT-PCR on CSF sample to identify the causative virus. What is the describe the enzyme correctly? RNA dependent RNA polymerase, not DNA dependent RNA polymerase. RNA dependent RNA polymerase. Which type of DNA repair mechanism involves the removal and replacement of DNA strand with the help of undamaged complementary strand as a template? Homologous recombination. நீங்க அந்த DNA ரிப்பேர்ல கொஞ்சம் கான்சென்ட்ரேட் பண்ணுங்க. என்னென்ன மெத்தட்ஸ் இருக்கு அண்ட் வாட் ஆர் த டைப்ஸ் யூ ஹேவ் டு நோ, பிகாஸ் நிறைய கொஸ்டின்ஸ் கேட்டிருக்காங்க. மெட்டபாலிக் சிண்ட்ரோம் நான் மார்னிங் சொன்னல, இதை நான் சொல்றேன்னு சொல்லிட்டு. மெட்டபாலிக் சிண்ட்ரோம் இன்க்ளூட்ஸ் 5: ஹை பிபி, ஹை குளுக்கோஸ், ஹை டிஜிஎல், low LDL and abdominal obesity. So these are the 5 factors which constitutes metabolic syndrome: high BP, high glucose, high TGL, low HDL, abdominal obesity. Next one, the acute intermittent porphyria is due to uroporphyrinogen 1 synthase defect. Congenital erythropoietic porphyria is due to deficiency of uroporphyrinogen 3 co-synthase. Decarboxylase deficiency causes porphyria cutanea tarda. Hereditary coproporphyria is due to coproporphyrinogen oxidase. Variegate porphyria is due to deficiency of protoporphyrinogen oxidase. Protoporphyria is due to deficiency of ferrochelatase. This kandipa ஞாபகம் இருக்காது. You have to paste it on your room and you have to memorize it. Okay va? Based on site of expression of the disease வந்து பார்ஃபேரியா வந்து ஹெப்பாட்டிக் எரித்ரோபைட்டிக் போத். எரித்ரோபைட்டிக் வந்து கஞ்சனிட்டல் எரித்ரோபார்ஃபேரியா அண்ட் எரித்ரோபைட்டிக் புரோட்டோபார்ஃபேரியா. இது எரித்ரோன்னு வர்றதெல்லாம் எரித்ரோபைட்டிக். ஹெப்பாட்டிக் வந்து அக்யூட் இன்டர்மிட்டன்ட் பார்ஃபேரியா. ஓகே, தென் வேரிகேட் பார்ஃபேரியா, தென் அக்யூட் இன்டர்மிட்டன்ட் பார்ஃபேரியா, வேரிகேட் பார்ஃபேரியாலாம் வந்து லிவர். எரித்ரோபைட்டிக் வந்து கஞ்சனிட்டல் எரித்ரோபைட்டிக் பார்ஃபேரியா அண்ட் எரித்ரோபைட்டிக் புரோட்டோபார்ஃபேரியா. பார்ஃபேரியா குட்டேரியா டாடா வந்து பார்ஃபேரியா குட்டேனியா டாடா வந்து போத். ஓகேவா? அக்யூட் நான் அக்யூட். எரித்ரோபைட்டிக் எல்லாமே நான் அக்யூட் குட்டேனியஸ்லாம். அப்புறம் போத் இருக்கிறதும் நான் அக்யூட்ல வருது. மீதி எல்லாமே வந்து அக்யூட்ல வருது. லிவர்ல வர்றது எல்லாமே அக்யூட்ல வருது. அக்யூட் இன்டர்மிட்டன்ட் பார்ஃபேரியா, வேரிகேட் பார்ஃபேரியா, hepatic porphyria. Okay, these are all will come under this one. Then I will say in CAG repeats. CAG repeats. Where the CAG repeats will occur? Any idea? நாலு இருக்குது. Huntington's disease. Huntington's disease. Huntington's disease தான் வந்து CAG repeats வரும். Muscle weakness, cognitive decline, involuntary movements, microsatellite instability, trinucleotide repeated sequence வந்து Huntington's diseaseல வரும். ஓகேவா? That is CAG repeats occurs in Huntington's disease. Okay. Okay, all of you do well. Concentrate more on your MCQs now. You have to read the questions and answers, correct answers. Read the questions and answer repeatedly. If you read the question answer, then only you will be able to tell you are able to mark your MCQs correctly. Okay, repeated reading, maximum number of MCQs you have to try to complete in a single day. You have to 1000 MCQs at least. Then only you will be able to. You have to revise 3 times at least before going to exam. So accordingly, you have to plan and do well in your exams. All the best for your performance, and all of you will clear your subjects. Okay, all the best.